Ladies and gentlemen, today we're confronting one of the health crises of our time, sarcopenia, the age-related loss of muscle that robs millions of strength, energy, independence, and lifespan. But what if I told you that food, not drugs, not surgeries, could be the most powerful weapon you have against this decline? Let's begin by understanding something critical. Sarcopenia is not just aging. It's a progressive loss of lean muscle mass and function driven by hormonal decline, mitochondrial decay, a chronic inflammation, and yes, nutrient deficiency. Your muscle is more than tissue. It's a metabolic engine, a glucose regulator, and a longevity organ. Preserving it is preserving your independence, your energy, your identity. And that begins with food, the right food. So here are the 7 essential foods backed by neuroscience and metabolic biology that can help you say goodbye to muscle loss and hello to renewed vitality. Sarcopenia, the age-related loss of muscle mass and function, is not simply the result getting older. It's the outcome of very specific biological processes that accelerate, if not actively addressed. At its core, sarcopenia involves the dysregulation of muscle protein turnover, a progressive imbalance where muscle breakdown, catabolism, begins to outpace muscle building anabolism. This breakdown accelerates due to a constellation of factors, most notably hormonal changes, mitochondrial dysfunction, chronic low-grade inflammation, inflammaging, and nutrient resistance. As people age, key anabolic hormones such as testosterone, growth hormone, and IGF-1 begin to decline. These hormones play a central role in muscle regeneration and repair. Without sufficient hormonal signaling, the muscle stem cells, known as satellites, have become less responsive, leading to impaired muscle recovery and regeneration after stress or injury. At the same time, aging mitochondria become less efficient, reducing ATP production and increasing the generation of reactive oxygen species, ROS, which further damages muscles at the cellular level. Additionally, muscle fibers shift from fast twitch type yank to slower twitch, dev type I, diminishing power, coordination, and reaction time, a transformation that's partially driven by denervation, where nerves withdraw from muscle units and the body fails to reinnervate them properly. Compounding this is a state of anabolic resistance, a condition where aged muscle does not respond as robustly to protein intake. This means that even when older individuals consume protein, their body doesn't efficiently convert those amino acids into new muscle tissue without specific interventions. Chronic systemic inflammation, often caused by poor diet, visceral fat accumulation, or gut dysbiosis, sends pro-inflammatory cytokines like TNF and IL-6 into the bloodstream, which blunt the body's anabolic response and accelerate tissue breakdown. And to this, the growing evidence that the gut microbiome influences muscle via the gut-muscle axis, and the picture becomes even more complex. Muscle loss, therefore, is not a localized issue. It is a full-body metabolic shift that involves immune signaling, hormonal regulation, neural inputs, and mitochondrial health. Muscle is not merely a mechanical system of fibers pulling on bones. It is a deeply integrated neurobiological structure governed by a continuous feedback loop between the nervous system and the muscular system. This integration begins in the motor cortex of the brain, where voluntary movement is initiated and flows down through the spinal cord to motor neurons that innervate skeletal muscle fibers. Every contraction, every stabilization, every fine motor control is orchestrated through this neuromuscular connection. With aging, however, this connection begins to degrade. The number of motor neurons declines, and existing neuromuscular junctions, the synapses where nerves meet muscles, become structurally unstable and less efficient. This degeneration leads to a phenomenon called motor unit remodeling, where fewer neurons are left to control larger groups of muscle fibers, reducing the precision and strength of movement and accelerating muscular atrophy. This breakdown in neuromuscular communication not only diminishes strength, but also impairs balance, coordination, and reflexes, which significantly increases the risk of falls and injury. Furthermore, the sensory feedback mechanisms that report muscle position, tension, and load back to the central nervous system become dulled, reducing the body's ability to self-correct during movement. The degradation of this feedback loop means that muscles are less likely to be stimulated appropriately, especially during low intensity or habitual activities, leading to underuse and further atrophy. On a biochemical level, neuromuscular disconnection reduces the release of acetylcholine, the neurotransmitter responsible for initiating muscle contraction. This decline is not just a symptom, but a contributor to muscle loss, because the absence of strong, consistent neural activation leads to decreased expression of genes involved in muscle maintenance, growth, and repair. Recent studies have shown that even in the absence of significant physical trauma, denervation can trigger a cascade of catabolic processes inside muscle fibers, shrinking them from within. The nervous system is not a passive player in muscular health. It is a central regulator that determines determines when and how muscle is built, maintained, or lost. Without robust neuromuscular signaling, muscles become functionally isolated, metabolically inefficient, and structurally vulnerable to wasting, regardless of protein intake or exercise. The timing of nutrient intake plays a pivotal role in the regulation of muscle protein synthesis, particularly in aging populations who experience blunted anabolic responses due to a phenomenon known as anabolic resistance. While total daily protein intake is important, emerging evidence suggests that when protein and other key nutrients are consumed can drastically influence the ability to maintain and build lean muscle. After a period of fasting, such as during sleep, muscle tissue becomes more receptive to amino acids, making the first meal of the day a powerful anabolic opportunity. Consuming a leucine-rich, protein-dense breakfast within an hour of waking aligns with the body's natural circadian rhythm and increases muscle protein synthesis more effectively than spreading protein intake thinly throughout the day. This is due to heightened insulin sensitivity and MTR pathway activation, which are more responsive in the morning and early afternoon hours. Furthermore, spacing protein doses strategically, typically 25 to 40 grams of high-quality protein every 3 to 4 hours, helps sustain an anabolic environment throughout the day while preventing extended catabolic windows where the body may begin to break down muscle tissue for energy. This is especially critical for older adults whose muscles do not respond robustly to smaller protein boluses. The post-exercise window also serves as a critical timing point. After resistance training or mechanical loading, muscle cells increase their sensitivity to amino acids and glucose uptake, amplifying the repair and rebuilding process. Ingesting protein with a moderate amount of carbohydrates within hydrate 30 to 60 minutes post-exercise has been shown to not only accelerate recovery, but also enhance long-term hypertrophy and strength gains. Evening timing plays its own unique role. The inclusion of slow digesting proteins like casein before sleep provides a steady stream of amino acids during the overnight fast when growth hormone secretion peaks and muscle repair processes are active. Missing these time-dependent opportunities can impair recovery and accelerate atrophy, particularly in older adults. The circadian clock influences nutrient metabolism through clock genes expressed in skeletal muscle, liver, and adipose tissue, meaning that nutrient timing is tightly linked to hormonal rhythms, insulin efficiency, and mitochondrial energy availability. Ignoring this synchronicity can mean that even the highest quality foods may be utilized inefficiently by the body. Strategic nutrient timing, therefore, is not just a performance hack for athletes. It is a biological imperative for anyone seeking to preserve muscle tissue, regulate metabolic health, and resist the degenerative cascade of sarcopenia. Hormonal windows and critical periods throughout the day when the body's internal biochemistry is most primed to support muscle maintenance, growth, and repair, especially as we age and experience natural declines in anabolic hormone production. Understanding how to leverage these hormonal rhythms is essential in the fight against sarcopenia, which is deeply influenced by diminishing levels of testosterone, estrogen, growth hormone, insulin like growth factor 1 and IGF-1, and even insulin itself. These hormones are not static. They fluctuate in a diurnal pattern that corresponds with circadian rhythms, physical activity, light exposure, and nutrient intake. For example, testosterone, a key anabolic hormone, reaches its peak levels in the early morning hours. Aligning protein intake and resistance training during this window can amplify the body's ability to synthesize muscle tissue and promote recovery. Growth hormone, another powerful anabolic driver, is secreted in pulsatile bursts with the most significant release occurring shortly after the onset of deep sleep. This means that preparing the body for high-quality sleep through nutrition, light management, and physical wind-down strategies can optimize muscle repair processes that are hormonally governed. Consuming a small amount of protein, particularly casein or collagen combined with glycine and magnesium, before bed can support this hormonal window, providing amino acid availability precisely when growth hormone levels surge. Additionally, IGF-1, which is stimulated both by growth hormone and insulin, plays a central role in muscle hypertrophy and satellite cell activation. IGF-1 sensitivity can be enhanced through resistance training, omega-3 fatty acids, and foods rich in zinc and vitamin D, all of which support the signaling cascade that leads to muscle regeneration. Insulin, though often viewed purely through the lens of glucose metabolism, is also a potent anabolic hormone when managed properly. After training or during periods of elevated insulin sensitivity, such as the morning or post-exercise, pairing carbohydrates with protein helps shuttle amino acids into muscle tissue more effectively. However, chronic hyperinsulinemia, often seen in older adults with sedentary lifestyles or metabolic dysfunction, leads to insulin resistance, which impairs this anabolic function and contributes to muscle loss. Thus, leveraging insulin's anabolic potential requires careful modulation of carbohydrate quality, timing, and physical activity. Cortisol, the body's primary stress hormone, must also be considered. Chronically elevated cortisol levels, particularly in the evening, can suppress testosterone and growth hormone production while promoting muscle breakdown. This makes stress management, circadian-aligned sleep, and light exposure essential components in maintaining an optimal hormonal environment for muscle retention. By strategically aligning food intake, exercise, and sleep with these hormonal peaks and valleys, individuals can create a metabolic landscape that supports muscle preservation and resists the catabolic forces of aging. The gut microbiome, a complex ecosystem of trillions of microorganisms residing primarily in the intestines, has emerged as a central player in not only digestion and immunity, but also in regulating skeletal muscle mass, strength, and metabolic function, a relationship known as the microbiome-muscle axis. This bidirectional system connects gastrointestinal health to muscle physiology through immune, metabolic, endocrine, and neural pathways. Creating a dynamic feedback loop becomes increasingly critical with age. As the gut microbiome shifts in composition over time, a process called dysbios, there is often a loss of microbial diversity and an overrepresentation of pro-inflammatory bacteria. This imbalance leads to increased intestinal permeability, or leaky gut, which allows lipopolysaccharides, LPS, and other inflammatory compounds to enter the bloodstream, triggering systemic inflammation that directly impairs muscle protein synthesis and accelerates muscle breakdown. At the same time, beneficial bacteria produce metabolites like short-chain fatty acids, SEO-Friendly, particularly butyrate, acetate, and propionate, which play vital roles in preserving muscle health. Butyrate, for instance, not only serves as a primary energy source for colon cells, but also possesses anti-inflammatory and epigenetic regulatory properties that influence gene expression related to muscle regeneration and mitochondrial function. SCFAs can also enhance insulin sensitivity, which is essential for muscle glucose uptake and anabolic signaling. A healthy gut microbiome supports the absorption of key nutrients that are foundational for muscle health, such as amino acids, magnesium, calcium, and B vitamins. It also influences appetite regulation and hormonal balance via gut-derived hormones like GLP-1, PY, and ghrelin, which affect satiety, metabolism, and the body's overall anabolic tone. Emerging research also suggests that the microbiome modulates levels of myostatin, a protein that inhibits muscle growth. Certain gut bacteria appear to suppress myostatin expression, allowing for more robust muscle protein synthesis, particularly when paired with resistance training and a protein-rich diet. Moreover, the microbiome plays a role in regulating the immune system's tone, which is critical for muscle recovery following exercise or injury. Chronic low-grade inflammation, commonly seen in older adults with dysbiosis, interfere with this recovery process, leading to incomplete repair, fibrosis, and long-term atrophy. Prebiotics, probiotics, and fermented foods can modulate the microbiome environment by encouraging the growth of beneficial bacterial strains like Lactobacillus and Bifidobacterium, which have been linked to improved muscle function and reduced inflammatory signaling. The microbiome's influence extends to the central nervous system via the gut-brain axis, which, through the vagus nerve and immune signaling molecules, can affect neuromuscular coordination, mood, and motivation, factors that indirectly influence physical activity and thus muscle health. Stress, sleep disruption, and poor dietary choices can all negatively affect the microbiome, leading to cascading effects on muscle metabolism. Therefore, nurturing the environment is not merely a digestive concern. It is a muscular, metabolic, and neurological imperative that plays an underappreciated yet powerful role in the prevention of sarcopenia. Behavioral neuroscience provides powerful insight into how habits, environmental cues, motivation, and brain chemistry intersect to shape not just our choices, but the biological outcomes of those choices, including the preservation or deterioration of muscle tissue. The brain is not simply a command center issuing orders to the body. It is a dynamic plastic organ that learns apps and reprograms behavior in response to reward, repetition, and internal biochemical states. At the core of this relationship is the dopaminergic system, which governs goal-directed behavior and habit formation. Dopamine release is not just about pleasure. It's about anticipation, motivation, and sustained effort toward outcomes, and it can be leveraged to build consistent behaviors that preserve muscle and reverse the drift toward sarcopenia. When behaviors like protein consumption, resistance training, and circadian-aligned eating are paired with immediate forms of reward, such as environmental priming, social reinforcement, or identity affirmation, they become more likely to transition from conscious effort into automatic routine, embedded within the basal ganglia's habit circuitry. The prefrontal cortex, responsible for executive function and long-term planning, must be engaged deliberately in older adults to override the inertia that comes with behavioral rigidity or motivational fatigue. By anchoring new habits to pre-existing routines, such as associating strength work with brushing teeth or linking morning protein intake to exposure to natural light. The brain forms durable neural pathways that reduce friction and promote consistency. Moreover, the circadian system, which regulates not only sleep-wake cycles but also hormone release, metabolic function, and cognitive performance, is tightly coupled with behavior. Light exposure, meal timing, and physical movement all send signals to the brain's suprachiasmatic nucleus (SCN), which then coordinates peripheral clocks in muscle, liver, and adipose tissue. When do behaviors align with circadian timing, such as skipping breakfast, eating late at night, or exercising at irregular hours? The brain interprets this as stress, activating cortisol and inflammatory cascades that inhibit muscle protein synthesis and accelerate breakdown. Behavioral neuroscience also highlights the role of interoception, or the brain's perception of internal bodily states, which influences how we interpret fatigue, hunger, and effort. Older adults may misinterpret anabolic signals as general discomfort or satiety, leading to underfeeding or movement avoidance unless these signals are retrained through mindful practice and cognitive reframing. Neuroplasticity, the brain's ability to reorganize itself through experience, does not disappear with age. It merely becomes more use-dependent. Therefore, behaviors that are practiced consistently, especially those that are emotionally salient or socially reinforced, create stronger and more durable connections in both motor and motivational circuits. Integrating muscle-preserving behaviors into daily life through cues, rewards, repetition, and identity alignment can shift the neurobiological baseline from passive degeneration to active maintenance. This means that the battle against sarcopenia is not only in the gym or the kitchen. It is won in the brain through the deliberate engineering of patterns, environments, and internal motivations that support long-term muscular resilience.